In the summer of 2008, in a cave in the Altai Mountains of southern Siberia, a Russian excavation team recovered a piece of bone about the size of a shirt button. It was the tip of a finger, the last joint of a little finger belonging to a child who had died somewhere around 40,000 years ago. Nobody was especially excited. The cave, Denisova Cave, named after a hermit said to have lived there in the 18th century, had been producing bones and stone tools for decades. The reasonable assumption was Neanderthal. Neanderthals had lived in that valley, and the fragment was far too small for its shape to tell anyone anything at all. The excavation there has been run for decades by Anatoly Derevyanko and Michael Shunkov of the Russian Academy of Sciences in Novosibirsk. Denisova Cave is not a spectacular sight to look at. It is a modest chamber in a limestone bluff above the Anhui River, with a hole in the roof that lets a column of daylight down onto the floor. What makes it valuable is unglamorous. It is cold, it is dry, and its layers have stayed undisturbed. Bone that would have turned to nothing in a warmer place has sat there, quietly, for tens of thousands of years. So the fragment was sent to a laboratory in Leipzig to have its DNA read. 2 years later, in December 2010, David Reich, Richard Green, Martin Kircher, and their colleagues published what came back in the journal Nature. The finger did not belong to a Neanderthal. It did not belong to a modern human either. And the living people who carry the most DNA from whoever it did belong to are not in Siberia. They are not anywhere near Siberia. They are roughly 8,000 kilometers away, across an entire continent and an open ocean, in the islands of the Western Pacific. This is the story of how a fingertip in a Siberian cave turned out to be the first physical trace of an entire kind of human that nobody knew had ever existed, and how the strongest evidence that they were real is not in a museum drawer at all. It is walking around, in the bodies of living people in New Guinea, in Bougainville, and in the Philippines. If you find these stories worth your time, subscribing genuinely helps, and stay to the end because there is something about the way this gets reported, a number you may have seen quoted about Pacific Islanders, that almost everyone reads backwards, including people who mean well. To understand why one finger bone caused so much trouble, we first need to understand the model it landed in. By 2008, the outline of human origins looked settled. Anatomically modern humans appear in Africa. Somewhere after about 60,000 years ago, a subset of that African population moves out across Eurasia and eventually everywhere else. On the way, they encounter other kinds of human already living there, populations descended from much earlier migrations out of Africa who had been evolving separately for hundreds of thousands of years. The best known of those are the Neanderthals. For most of the 20th century, whether our ancestors and those other humans had children together was an argument you could not settle. You could measure skulls. You could argue about brow ridges. You could not read the DNA of someone who died 40,000 years ago because there was almost none of it left to read. That changed in May 2010. Richard Green, Svante Pabo, and a large international team published a draft genome of the Neanderthal in the journal Science, assembled from bone powder taken from 3 individuals in a cave in Croatia. The result was not ambiguous. People whose ancestry lies outside sub-Saharan Africa carry a small but real fraction of Neanderthal DNA. The 2010 estimates put it at roughly 1 to 4%. It is worth sitting with what that actually means. It means the encounter was not just contact. It means that somewhere in the Middle East, 40 or 50,000 years ago, 2 populations that had been apart for hundreds of thousands of years met and had children, and those children survived and had children of their own, and the line held all the way down to the present. Most people watching this are carrying the evidence of that in their cells right now. It is worth being clear about how hard that read is, because it explains almost everything about the pace of this field. DNA begins breaking apart the moment an organism dies. In an old bone, what survives is not a genome. It is fragments, most of them shorter than 100 letters, and the overwhelming majority of what you pull out of the powder is not the individual at all. It is bacteria that colonize the bone in the ground, plus whatever DNA has been shed onto it by every living person who has handled it since. So the work is mostly a filtering problem. You are separating a faint, damaged, ancient signal from a much louder modern contaminant, and the tell is chemistry. Real ancient DNA carries a specific pattern of decay at the ends of its fragments that fresh DNA does not have. That was the state of the science when the finger bone arrived in Leipzig. The first signal came before the full genome. In March 2010, Johannes Krause and colleagues published the mitochondrial DNA from the fragment, also in Nature. Mitochondrial DNA is a small, separate loop of genetic material passed down the maternal line, and there are many copies of it in every cell, which makes it the easiest thing to recover from an old bone. It did not match modern humans. It did not match Neanderthals either. On the maternal line alone, this individual's lineage appeared to have separated from both deep in the past. Mitochondrial DNA on its own is not enough to name a population. It is one thread out of the whole cloth, and a single unusual thread can have unusual explanations. So the team kept going and pushed for the nuclear genome, the full set of chromosomes inherited from both parents. They got it to about 1.9-fold coverage. In plain terms, that is a thin read. On average, each position in the genome was seen roughly twice. It is nothing like the depth used in medical sequencing today. But it was enough. The individual was female. Her population shared a common origin with Neanderthals, a sister group splitting from them long before either met us. But she was not a Neanderthal, and the differences were not small. A molar found in the same cave carried nearly the same mitochondrial genome, and that tooth was large and archaic in a way that matched neither Neanderthals nor modern humans. The team named the population after the cave, Denisovans. Consider the position that puts you in. An entire kind of human being, one that lived alongside our ancestors, had been discovered and described, and it had been done without a skull, without a jaw, without a skeleton. It had been done from a fingertip and a tooth. They had the handwriting before they had the writer. That alone would have been the finding of a career. But there was more. Because once you have a genome, you can ask the obvious question: is any of this still around? The team compared the Denisovan sequence against 53 present-day human populations. What they were looking for was signal, living people whose DNA sat unusually close to this archaic individual. The population split into 3 groups. The sub-Saharan African populations formed one. A second group held 44 populations from across Europe, Asia, and the Americas, plus one North African group, essentially everybody else. And then there was a third group, standing apart from all of them. That third group was 2 populations, Papuans from New Guinea and islanders from Bougainville. The paper's estimate was that 4 to 6% of the Melanesian genome derives from Denisovans. The central figure in the analysis was 4.8%, plus or minus half a percentage point. Stack that on top of the Neanderthal component, and as much as 7.4% of a Melanesian genome traces to archaic human populations. To put the scale of that in perspective, it helps to know what a percentage of a genome physically looks like. Archaic ancestry does not sit in one place. It is scattered. Hundreds of separate stretches of sequence distributed across the chromosomes. Each one a surviving piece of a much larger inheritance that has been broken up and thinned by recombination over roughly 2,000 generations. Nobody carries a Denisovan chromosome. What people carry are the pieces that were useful enough or lucky enough to still be there. And Melanesia is not a small or marginal place. It is the arc of islands running east from New Guinea, the Bismarck Archipelago, the Solomon Islands, Vanuatu, New Caledonia, Fiji. And New Guinea itself is the second largest island on Earth, home to well over 1,000 distinct languages. Bougainville, which appears in the 2010 analysis, sits at the western end of the Solomons. These are among the longest continuously inhabited places outside Africa. Now look at a map and the problem becomes obvious. Denisova Cave sits in the Altai Mountains in continental Siberia, at the meeting point of Russia, Kazakhstan, Mongolia, and China. New Guinea is on the far side of Asia, past the Himalayas, past mainland Southeast Asia, past a stretch of deep water that has never in human history been dry land. The two places are about 8,000 kilometers apart, and the people living closest to the cave, Siberians, Mongolians, Central Asians, showed essentially none of this ancestry. The signal skipped everyone in between. The explanation the team reached is the one that has held up. Denisovans were never a Siberian species. They were, in the paper's own suggestion, likely widespread across Asia during the late Pleistocene. Denisova Cave is not their homeland. It is their freezer. It is simply the one place we have found where the conditions were cold and stable enough for their DNA to survive long enough for us to read it. Somewhere in the warm, humid south, where bone dissolves and DNA falls apart within centuries, a whole population lived and moved and met the ancestors of Melanesians, and left almost nothing behind but what it put into them. Which means the 4 to 6% is not a curiosity. It is the primary evidence. For most of what we know about Denisovans, the living are the archive. But even this was not the full story. In April 2019, Guy Jacobs, Murray Cox, and a large team published in the journal Cell. They had sequenced 161 new genomes, spanning 14 island groups across island Southeast Asia and New Guinea. The question was straightforward. Now that we can find Denisovan stretches inside living people, do they all look like they came from the same source? They did not. The Denisovan segments in Papuan genomes fall into two groups so different from one another that the populations they came from must have separated more than 350,000 years ago. Both are also distinct from the individual in Denisova Cave. And a third, separate Denisovan lineage shows up in modern East Asians. Think about what that means for a moment. Not one lost population. At least 3 spread across Asia, isolated from each other for longer than our own species has existed, each of them meeting modern humans separately. There is a further detail in that paper, and it is the one that changes the picture most. The evidence suggests that one of those admixture events happened east of the Wallace Line. The Wallace Line is a boundary that runs through the Indonesian archipelago, separating the shallow continental shelf of Asia from the deep water beyond it. West of it, during the Ice Ages, islands joined into a single landmass you could walk across. East of it, they never did. The channels there stayed open and deep in every era. To be east of the Wallace Line, someone had to cross open water. Denisovans, or a population closely related to them, appear to have been over there, on islands you cannot reach on foot, and to have still been there late enough to meet arriving modern humans near the end of the Pleistocene. And the deeper the sequencing went, the stranger the distribution became. In August 2021, Maximilian Lorena, Matthias Jakobsen, and their collaborators published in Current Biology, working with communities across the Philippines. It was a genuinely large undertaking, around 2.3 million genetic markers from 1,107 individuals across 118 distinct Philippine ethnic groups. The record for the highest Denisovan ancestry on Earth did not belong to Papua New Guinea. It belonged to the Ayta Magbukon, an indigenous community in Bataan on the island of Luzon. Their Denisovan ancestry runs roughly 30 to 40% higher than that of Papuans and Aboriginal Australians. Once the researchers accounted for more recent mixing with East Asian populations, the gap widened to as much as 46%. The Ayta Magbukon are one of the peoples historically grouped under the label Negrito, an exonym applied by Spanish colonists, still used as a technical term in the genetics literature, and not a name these communities chose for themselves. Where it matters here, the specific community is what counts. And the reason they hold the record is worth holding on to, because we are going to come back to it. Keep it in mind. While the geneticists were finding Denisovans inside living people, someone finally found a piece of one above ground. and it turned out it had been sitting in a drawer the whole time. In 1980, a Buddhist monk was in Baishia Karst Cave, a sacred site in Xiahe County in Gansu Province on the northeastern edge of the Tibetan Plateau. The cave floor is more than 3,000 meters above sea level. He found the right half of a heavy human jaw with 2 large molars still attached and passed it on. It went to Lanzhou University, where it stayed, essentially unstudied, for close to 40 years. In May 2019, Fahu Chen, Dongju Zhang, and their colleagues published what it was in Nature. No DNA had survived in it, so instead they read the collagen proteins inside one of the molars, a technique that works on material far too old and too degraded for genetics, and the protein sequences matched Denisovans. A crust of mineral had built up on the surface of the jaw. Dating that crust gave a minimum age of about 160,000 years. It was the first confirmed Denisovan found anywhere outside Denisova Cave. And it was found at high altitude, in thin air, more than 100,000 years before modern humans reached the Tibetan Plateau. If that detail sounds familiar, it should. The gene that lets modern Tibetans live comfortably at altitude, EPAS1, was traced by Emilia Huerta Sanchez and colleagues in 2014 to a Denisovan source. The jaw in the drawer is, in effect, the body that goes with that gene. There is one more fossil worth naming because it is the most human thing in this entire story. In 2018, Vivian Slon, Svante Pabo, and colleagues published the genome of a bone fragment from Denisova Cave, catalogued as Denisova 11. It came from a girl who was at least 13 years old when she died. Her mother was a Neanderthal. Her father was a Denisovan. Not a distant ancestor from either group, her actual parents. She was a first-generation child of 2 populations that had been separate for hundreds of thousands of years. And we found her on the first real attempt, which suggests that in that cave, in that period, this was not extraordinary. The picture that emerges is not a set of separate species keeping to themselves. It is a crowded, overlapping world. Island Southeast Asia, in particular, was full of other kinds of human. Homo floresiensis on Flores, and Homo luzonensis, described by Florent Destois and colleagues in 2019 from Callao Cave. On Luzon, the same island where the Ayta Magbukon live today. Now consider the inventory. 16 years after Denisovans were named, the physical remains attributed to them amount to a fingertip, a handful of teeth, a partial jaw, some bone fragments, a rib, and DNA recovered directly from cave sediment. There is no Denisovan skull. There is no Denisovan skeleton. Nobody knows what their face looked like, and any picture you have seen of one is an artist's guess. We know them almost entirely from the margins of other people's genomes. That was the analogy at the start, and it holds all the way down. We found the handwriting before we found the writer. Everything substantial we know about an entire branch of humanity, where they lived, how many groups there were, how long they lasted, that they crossed open water, has been reconstructed from annotations they left in a text that is still being read. The text is living people. So what does the finding actually amount to? It means the shape everyone was taught is wrong in a specific way. A family tree branches, and the branches move apart, and they never touch again. That is not what happened. Lineages separated for 300,000 years came back together and merged, more than once, in more than one place, with more than one group. The structure is not a tree. It is a braid. And this is where the number gets read backwards. 4 to 6% archaic DNA in Melanesians gets quoted constantly, and it is almost always framed as though it means those populations are somehow older or closer to the past or less finished than everyone else. That reading is not a matter of opinion. It is a factual error, and the research itself is what corrects it. Start with the obvious. Every living human lineage is exactly the same age. Every person alive descends from an unbroken chain reaching back to the same origin. Nobody's line is longer. Nobody stopped early. Then take the ancestry itself. Almost everyone outside sub-Saharan Africa carries archaic DNA. What differs is not how much history someone has. It is which other population their ancestors happened to run into on the road they took. The people who went east through island Southeast Asia met Denisovans. The people who went north and west met Neanderthals. That is a difference of root, not of rank. And then there is the Ayta Magbukon, which is the part I asked you to hold on to. They hold the world record for Denisovan ancestry, not because they accumulated more of it. They hold it because they mixed less with a later arriving population that carried less of it, so less of theirs was diluted. The highest figure on Earth is a measure of who did not arrive. It is a demographic accident recorded in a genome. Read carefully, the single most extreme number in this entire field turns out to be evidence against the idea of ranking populations at all. What the Denisovan story actually records is contact. It is the genetic memory of ordinary human beings meeting other human beings, in Siberia, in the Philippines, on islands past the Wallace Line, and having families. The thing that makes Melanesian and Philippine genomes distinctive is the same thing that makes them not separate. Somebody's ancestors met somebody else's and stayed. If you have ever taken a commercial ancestry test and found a small percentage you could not account for, an archaic fraction, a trace component, some line that did not match the story your family tells, this is part of that world. Those numbers are estimates against reference panels, and they move as the panels improve. But underneath the rounding, they are pointing at something real. A great many meetings, most of which nobody recorded, and some of which involved people who were not quite us. The population from Denisova Cave is gone. No group alive today is Denisovan, and none is descended from them alone. But they are not exactly absent either. They are annotations in the margin, in the bodies of people in New Guinea, in Bougainville, in Bataan. Sequences still doing quiet work in immune systems and in the way blood handles thin air, 40,000 years after the last of them died. We found the handwriting before we found the writer. We still have not found the writer. And we may never need to, because the book is still open, and people are still reading from it. Thanks for watching to the end of this one.